Welcome back to the course Fundamentals of General Immunology, and we are understanding the mucosal immunity and mucosal immune response. So we kind of looked into all the details of the mucosal system, particularly the gut epithelium, the respiratory epithelium, genitourinary tract. We also looked into how these specific kind of tolerance are coming into picture when it comes to oral tolerance. We earlier looked into central and peripheral tolerance, which are basically the tolerance which are coming when we are having a self-reacting T cell or B cell population, how they are getting eliminated. Those are called central and peripheral tolerance. But then when we are taking any food particle, then we are eliciting some kind of tolerance against those food particles. We are not raising any immune response against those food particles. So those are coming under oral tolerance. So today we'll try to understand this IgA response, particularly the mucosal immunoglobulin response, which are coming into the mucosal epithelium, as well as some of the clinical relevance of tolerance, like how we can utilize this tolerance towards the usefulness for the human medicine or the human therapeutics. So let's try to move on and see that what are the important aspects of IgA secretions actually, or maybe some other immunoglobulin secretions. So there is a concept of mounting an IgA response when we come to a gut metabolite specific biology. So we'll try to understand what is the impact of gut metabolites on specific IgA response in the gut. So say these are the small intestinal area where food is coming, and once the food is digested, then we know that the food particles are, say, containing different kind of dietary fibers and also the microbiota which are present in the alimentary canal. These dietary fibers can convert into very important molecules which are called short chain fatty acids. And these short chain fatty acid might be of different types. It could be butyrate, acetate, or propionate. So these are some of the small or the short chain fatty acid varieties which are available when we take a dietary fiber into our food. Now these butyrate and acetate and propionate can modulate the immune response differently. Now this SCFA can do other activity as well. So let's try to focus on what they are doing in terms of the different kind of immunity in terms of dendritic cell. So small chain fatty acid can cause dendritic cell development and regulatory function by upregulating GPR109A. So GPR is actually G protein coupled receptor. So these GPR, different variety of GPR, for example, the GPR109 is available or expressed in dendritic cell, while GPR43 is expressed in T regulatory cell population. So they can upregulate the FOX3 induction and also T regulatory cell differentiation and expansion, and conversion of T regulatory cell into something which we call the T follicular helper cell. So this is the follicular helper cell which are coming into picture by the transition of T regulatory cell. So essentially the short chain fatty acids are basically modulating the dendritic cell by its development, by expressing some of the receptor on the cell surface. They are also modulating the T regulatory cell population in the lamina propria area, where they are modulating the expression of a very specific kind of protein called FOXP3. Along with that, they also regulate the follicular helper T cell population in the area which is very close to the gut wall. Now these acetate particularly are known to, say, inhibit. So this is a sign of inhibition. So this can inhibit the HDAC. HDAC is actually histone deacetylase, which can remove the acetyl group from the histone to destabilize the histone molecule. This is very important protein when it comes to gene regulation in case of the human beings and also in other animals. So HDAC regulation is largely studied in cancer biology as well as other molecular aspect of the cell system. So this is a very critical way it is regulating the immune cells as well. So HDAC is getting inhibited by acetate molecules which are coming from short chain fatty acids. Apart from that, we know that T regulatory cells are producing TGF beta and IL-10, and this TGF beta and IL-10 can modulate the expression of IgA molecules. Now these are basically cells which are naive cell which are not expressing anything. So these are IgM and IgD positive. So there are two immunoglobulin IgM and IgD are expressed on the cell surface. Now these are causing a class switch phenomena. So this is a class switch recombination that can happen in the B cell population, which can shift it to IgA production, and these IgA will essentially getting into circulation to nullify the pathogens and other things coming into the alimentary canal. So basically there are two ways. They can regulate the T regulatory cell population, and also they can modulate the class switching phenomena in the B cell in order to generate more and more IgA molecules. Apart from that, they can also do TGF beta and IL-10 production in the system. One important aspect about the IgA production in the gut is regarding this microbiota produced ATP. Now this microbiota produced ATP is known to modulate the expression of a very specific purinergic receptor on the follicular helper T cell population, which can inhibit basically the biology of T helper cell in order to produce other cytokines and immune response. So basically this can sequester the expression of the T helper cell biology in the follicular area. Essentially what we are getting is these IgA producing plasma cells which are coming into picture after these processes to secrete more and more IgA secretion in the epithelium. So these are basically the aspects which are related to both FOXP3 positive cells, where the T regulatory cell population is coming into the gut wall, and also since we need to neutralize the bacteria or any pathogen in the gut, we need a secretory immunoglobulin A molecules. So these are known to produce IgA into circulation to control the infection and all. So this is about how these gut metabolites are controlling the IgA population or IgA secretion in the gut wall. Let's try to see what else is happening when we are engulfing the short chain fatty acid. Now short chain fatty acid, as we understand that these plasma cell will come into circulation and start secreting immunoglobulin A, which has a dimeric form. Remember, we always write the secretory form of immunoglobulin A, which is always in the dimer. So this is actually a dimeric form. So this is one IgA and this is other IgA molecule which are joined by the J chain. So this is a secreted form of the immunoglobulin A molecules. So these are basically neutralizing this pathogen which are coming in the gut wall. So these are very important. Otherwise, the short chain fatty acid can also cause a systemic immune response by doing a class switching phenomena to produce more of IgG molecules. So we can see that, you know, naive B cell population which are expressing IgM and IgD, they can also go class switching recombination, and after class switching recombination we can see that production of IgG is coming into picture. So now these cells will go into circulation to control the expression and the secretion of IgG as well. So essentially these are typical form of the mucosal IgA and IgG production where the things are getting neutralized by few things, and one such very important thing is short chain fatty acid. So we can remember the short chain fatty acid does modulate lot of different immune molecules in the gut epithelial wall. So this is about how IgA is produced in the gut lumen. Let's try to understand some very critical thing about IgA and IgG as well. We can also look into IgD later part, but particularly in the females reproductive system, this is very important that in the female reproductive system, the pregnant females are transferring the IgG across placenta to the newborn or the growing fetus. So essentially, the transfer of circulating maternal IgG across the placenta is very critical phenomena to control any sort of infection in the growing fetus. So this has happened by multiple different proteins which are available in the body. So IgG circulating in the mother's body can pass through the fetus. So this is a fetal blood circulation, and this is mother blood. Mother and fetus is connected through this very specific structure called placenta, and this placenta is containing various kinds of cells, and one such cell is called syncytio trophoblast. Now trophoblast stands for any cell which can provide nutrition. Trophoblast stands for nutritional cells which can provide nutrition to any other cell. Syncytio stands for multinucleated. So if there are two cells and these cells are fused to form a multinucleated condition, we simply call it syncytia. So we call it syncytia. So syncytia is actually a multinucleated condition. So these are actually present in the villi of placenta, which are bathed through the maternal blood. Now this blood having this kind of a syncytio trophoblast can bind to this FcRn. FcRn is very specifically called neonatal Fc receptor. These are specific protein that can bind to the constant change of the immunoglobulin and can avoid its degradation through the endosomal pH. So basically it's a very protective mechanism that a female body can develop. So this FcRn is very specific protein that helps in the transfer of IgG molecule to fetus from the mother's blood. So this can bind to FcRn specific immunoglobulin G is now not able to degrade through the acidic pH because it is kind of protecting it from the degradation. It's a kind of a salvage receptor. People used to call it salvage receptor because it can help to sustain the IgG into the lumen, and then they can pass through this into the outer surface, which is called cytotrophoblast. Again, trophoblast is one which can provide nutrition. Cyto stands for cell. So these are the first and outer lining of the fetal membrane which contains this kind of a passage. So these can transcytosed and goes to the fetal endothelial cell. So remember that IgG is passively transferred to the fetus from the mother blood via placenta, and the molecule which helps in the transmission of IgG from the maternal to the fetus side is FcRn, which is called neonatal Fc receptor. Any deviation, anything which is happening in the architect of the FcRn or any malnutrition or anything in FcRn can cause the deviation of immune response to the young ones or the growing fetus in the mother's body. So this kind of immunity is very well studied when it comes to the different trimester of pregnancy. So this is all about how this IgG is transferred from mother's body to the fetus in placenta. Let's try to understand what happening after birth. So this is before birth, and after birth the mother is feeding through the breast milk. So the breastfeeding can also provide IgG and IgA to the fetus because their immune response, the young one's immune response, is not developed yet. So once they are born, they are susceptible for many virus, bacteria or parasitic infection. So the circulating IgG can pass through the breast tissue, again through the FcRn receptor, and then also the mother's gut, which are actually gut associated lymphoid tissue, can generate the IgA, which we have understood. So IgA produced from the gut can also passively goes to the breast tissue, and from the milk the IgG and the polymeric form of the IgM, the secretory form of the IgM along with the secretory form of the IgA which are coming in the circulation can pass through the fetus, and they can control the fetus with many kind of inflammatory processes, any kind of enterocolitis, that means any kind of inflammation that's happening in the intestine or colon, which is the large intestinal part. And also it helps in the immune maturation because it can sensitize the body in order to respond to an immunoglobulin. So this is very critically when it comes to IgG and IgA. So remember that the milk and the placenta is kind of a passage transferring the immunity, passive immunity. So that's not an active. So this is a ready-made form of the immunity which is transferred to the fetus or the young one in order to support them from any infection that's coming in from the environment, because simply their immune response is not developed yet. So now move on to a little bit about IgD, which is very less explored molecule, but still this is biologically very relevant. So let's try to understand what is happening when the fetus is grown up. So once we are looked into the IgG secretion in the fetus in the mother uterus, then after the birth, and finally what is happening when they are grown up. The mucosal immune response towards the IgD is very important, and that's important for aerodigestive tract. That means especially in the respiratory as well as digestive system. So these are again happening in the germinal center, mostly in the germinal center, and because of the somatic hypermutation and class switch recombination, the naive IgM and IgD containing B cell can switch themselves to produce an IgD from the system. And once IgD is produced, they can go and fight to the pathogen by moving out by transcytosis. So these are coming into the circulation. Again, they can come out in the circulation, and again they can do some other function as well. And one such function is they can suppress the activated basophil to release some of the allergic mediators, such as they can help in the mucosal defense by secreting antimicrobial peptides. They can recruit the immune cells for inflammation specific cytokines. So basically these are actually important when they bind to the CD44 and galactin-9 receptor. So IgD can dock with galactin receptor present in the basophil. So these are actually function of IgD. So IgD can go into the circulation and can bind to the commensal and pathogenic microorganism in the gut wall in order to neutralize them, and of course they can do some kind of a function. And of course we can also see that the basophil which are activated or say sensitized with IgD can do multi-function, where they can also do a B cell activation by BAF recruitment. So they can do a BAF recruitment, they can do IL-4, IL-5 and IL-13 generation. So basically these are Th2 immune response which are important for parasitic infection, which we have discussed in the earlier classes as well. They can also do kind of an subtype switching of Ig1, Ig4 and IgE parasitic aspect and antimicrobial. So there could be multiple function of IgD, but one thing which we need to remember here is these are very specifically mucosal immune system, and the somatic hypermutation is required in order to do a class switching phenomena where an IgM-D containing cell can move out to produce an IgD only. So these are double positive to a single positive cell population where they can secrete out only D and M becomes silent in this case. So we can see a variety of this class switch recombination phenomena. In the earlier cases we see that IgM and D containing cell are basically producing IgG. So there is a class switching entirely in a different class. Here we have double positive cells such as IgM and D, where one is getting silenced and the other is getting expressed in the large quantity so as to secrete out into the gut lumen. So this is all about the IgM and IgD along with IgG production in the gut as well as in mucosal epithelium and their relevance in terms of human aspects. Now we will try to see some of the things related to tolerance which we have discussed, that how we can utilize the concept of tolerance to do some clinical relevance in terms of treatment in human medicine. So we kind of understood that tolerance can be induced when we are taking a dietary intake. So when we have a more dietary intake, so what we understood that more and more peptides or kind of a dietary antigen can be loaded to the antigen presenting cell via MSC molecules. and this can cause more and more TCR to come into sight. So essentially, more and more TCR can cause the energy, so functional inactivation of the T cell. Also, the expression of the death domain, particularly CD95 and 95 ligand specific interaction, can cause a specific apoptosis of the cell. So basically, these are possible when we have a very large quantity, high dose specific antigen coming into picture. So this can cause high dose tolerance, means there could be a tolerance when we have a very high dose of the antigen, which can cause basically energy kind of process, and also there could be an apoptosis where there is no immune response. While in the other case, we have a low dose tolerance where the simple IL-10 and IgA beta can secrete and then can do upregulation of T regulatory cell population, and also they can suppress the effector T cell population as well. So essentially, the low dose and the high dose tolerance are doing differently, but the whole function is to produce and suppression. Now, what is the take home message is we know how to create a tolerance. So that means when we have a dietary intake of the antigen, basically that antigen won't mount an immune response in the human body. So say if I have an antigen which is going through the oral cavity, say if we ingest that antigen, probably they may not mount an immune response in a proper way, but when we inoculate the same antigen through the body cavity, we can mount an immune response. Like we can give an analogy and understand it in a different form. Suppose we are taking, say, chapati or any kind of dietary fiber. When we take through mouth, it won't raise an immune response. But when we grind it out, and if we give it by subcut or intramuscular injection, it may lead to allergic inflammation or any kind of reaction, because that's an unnatural route. So the oral is basically a way to attenuate the immune response, so down the immune response, and that is what you can use it to find out the way to treat some of the diseases. And few of the examples are inflammatory bowel diseases. So in inflammatory bowel diseases, now this is coming up in a very large scale in Indian population as well. People are suffering from inflammatory bowel diseases when the acidic pH of the alimentary canal is causing disruption of the epithelial layer of the intestine. They are not able to, say, digest the thing properly. There could be an infection, there could be multiple cytokines which are coming into picture, and also this can lead to say gastroesophageal reflex disorder by various ways. It can also cause infection of other parasite or other bacterial pathogen, and finally essentially what is required is basically T regulatory cell population. So this is lost, IgA production is lost, and the gut homeostasis is lost because the bacteria is no more available to maintain the proper pH and proper homeostatic atmosphere of the gut. Now this can lead to, say, deviation of the complete gut homeostasis and multiple disease condition, because there is an impaired epithelial barrier. So a barrier is breached. There could be secretion of multiple cytokines which can cause inflammation. So these are called pro-inflammatory cytokines such as TNF alpha, and this can make a system susceptible. Now this is something which is related to gut. So if there is a relation of gut involved here, how we can do this whole process and see that if we can still treat this disease with using tolerance mechanism into picture. So let's try to see what else we can do and what are the things available right now in research aspect wise that can help us to fight against inflammatory bowel disease or some of the autoimmune diseases. So let's try to understand this is in the form of an example. So suppose if we inoculate a mice with an antigen, suppose this antigen is a protein and we inoculate this by subcutaneous route. Subcut is just below the skin. So if you inoculate just below the skin, it will raise a very specific immune response after, say, 22 to 24 days. So generally after 3 weeks we see a huge immune response generated in these mice which are inoculated with that antigen. We can also inoculate antigen with adjuvant, which are, say, molecules which are given along with the antigen to boost an immune response. Adjuvants are basically a booster molecule which can give some kind of an uphold so that the antigen can mount a better immune response as compared to antigen alone. So sometimes adjuvants are given. There are other classification of adjuvants such as Freon's complete and incomplete adjuvants, which we'll see when we'll discuss the vaccine part in our final touch for this course. Now just see the scenario. So here we are raising an immune response which is very high immune response when we are giving this antigen through subcut route. Now what will happen when we change the antigen from the oral route? So what we understood that when we give the same thing through the oral, we won't get an immune response to a better extent. So same thing when we take this antigen and feed the mice through oral route, and then we inoculate this antigen by the subcutaneous route, we don't see the immune response. So that's causing tolerance. Same thing if we take the T cell from this individual and transfer this T cell also to a different individual, they can lead to tolerance. What does it mean? Is that once we are feeding that antigen, it is not raising an immune response. So the immune response which was raised here is not produced here. That means the T cell which are involved in this can be utilized for doing some function. And this T regulatory cell, when they are transferred into some other mice, they can also cause tolerance. That means if we take out this T regulatory cell, we can reduce the immune response. So this is what the concept is. We can make this as a model, and once we make this model, we can have a T regulatory cell coming out from this mice which are kind of tolerant, and this T regulatory cell can be transferred into the patient in order to reduce the immune response, because that will also create the tolerance, by the way. So how this concept is useful? This concept is useful in other way. For example, suppose this mice is suffering from autoimmune disease. So this mouse or a breed of mice, for example, are raising an immune response against any kind of antigen. Now to reduce this immune response, we can transfer this T regulatory cell to these mice in order to reduce the immune response, and therefore we can treat the mice from autoimmune disease condition. So mice is a very good model where we can study this kind of tolerance and some of the autoimmune related disease model. There could be other disease condition where different model system has been used. These are the diseases such as experimental autoimmune encephalomyelitis, which is actually a human disease counterpart where we study the multiple sclerosis. So these are in the mice model, and this is for the human. So at least what research we are doing in the animal model can be reciprocated for the human disease aspect. So multiple sclerosis is one such condition where the neurons are getting damaged because of the immune response against specific proteins. So then we have collagen induced arthritis, adjuvant based induced arthritis. Again, these are the inflammation of joint, and we can use the result of these study to rheumatoid arthritis, which is again a kind of autoimmune disorder in case of human being, where the antibodies are generated and can damage the joints. Uveitis is actually the inflammation of eye, which is again a model for our own body sometimes producing an immune response against some of the proteins in the eyeball. So those are called uveitis. Then experimental autoimmune myasthenia gravis, again the muscle specific disorder. This could be used to treat some of the patients who are suffering from myasthenia gravis. Diabetes type 1 and type where we can use some of the model in the mice where we can have an obese mice and follow some of the things where the immune cells are produced against the pancreatic cell, and therefore they won't mount a insulin specific response and won't be able to downregulate the glucose concentration in the blood. So we can use those to treat diabetes as well. We have a model for stroke as well. We have a model for atherosclerosis where the LDL, which is a low density lipoprotein receptor, which is absent, and then we can try to use those as a model to see that whether the result of those can be utilized for the human disease condition. Tissue transplant, of course, we can use the MSC specific biology when we use animal as a model and see if we can reciprocate those into a human being. So there are a lot of things which are of clinical relevance when it comes to tolerance, because we use the autoimmunity specific biology to fight against the disease as well when we transfer the T regulatory cells to those individuals. Now let's see another way, same thing in a different form, called lymphocyte reprogramming. So we can reprogram the T regulatory cell, we can reprogram the T cells in order to generate more and more T regulatory cells. So what we could do is we can transfer the antigen with our own host molecules. For example, we can use the splenocyte to couple the antigen, we can use the MHC molecule to couple the peptides, we can use the RBC to couple the antigen, and we can use some of the small particle, synthetic particle, to coat the antigen. Basically, these all antigens are using some of the molecules which are non-immunogenic in nature or the human specific way, so that the APC can present this and can sensitize the T helper cell or T cell in order to produce different kinds of cytokines, and these can ultimately lead to T regulatory cell upregulation and also the energy of the activated T cell population. So some of the T helper cell or the cytotoxic T cell population can be nullified by functional inactivation. They can downregulate the effector T cell population and can also upregulate the T regulatory cell population. So scientists are trying to do some of the new ways so that they can generate this T regulatory cell in order to transfer it to a patient who are suffering from autoimmune disorder. So this is some of the way people are trying to code the antigen using the self molecules. So RBC is self, splenocyte is self, these loaded peptide particles, synthetic particles, are non-immunogenic, MHC molecules are also self. So we can't see some of the immune response coming out because we can utilize the MHC molecule to load the peptides. So this is about lymphocyte reprogramming. We can program lymphocyte differently in order to upregulate the T regulatory cell population. So this is what is there in lymphocyte reprogramming. And finally, something called bystander suppression. Now bystander suppression, we are quoting here just an example of a condition where the myelin basic protein is MBP here. The myelin basic protein is actually causing a specific kind of reaction in the neurons. So neurons are getting problematic, say inflammation. Neurons are getting inflamed by this specific kind of a protein which are expressed, called myelin oligodendrocyte glycoprotein. So these are there, and against this protein the autoreactive T cells are produced, and therefore these autoreactive T cells can destroy the neurons. So that means these are our self protein, and because of this protein these autoreactive T cells can cause the destruction of the neuron, and therefore we can start getting a lot of symptoms against this. What we could do some kind of a sensitization. So we can use the myelin basic protein, which is actually the part. So this can be sensitized to the antigen presenting cell. Antigen presenting cell can present it to the T cell population, and essentially we can cause high activation of the T regulatory cell. Now these T regulatory cells are very specific against this myelin basic protein, MBP, and this T regulatory cell, which are very specific for myelin basic protein, can be given to the patient. Now since these T regulatory cells are regulating this myelin basic protein, so this can suppress the activity of that. So the T regulatory cell population can downregulate the immune response which are generated against this MOG, and basically we can treat the patient with this pre-sensitized T regulatory cell against myelin basic protein. So this is actually a bystander effect. So we are trying to use the autoreactive, we are trying to suppress the autoreactive T cell by a specific T regulatory cell which are sensitized with the myelin basic protein. So this is the way we are trying to move forward to treat some of the autoimmune disorder, because this is autoimmune disease, because immunity is generated against the self cell, particularly the neurons, and if we damage the neuron, we see a lot of complications, and therefore we can use the counter strategy to do a suppression by using the T regulatory cell population from the host cell. So this is all about the tolerance, the induction of IgA and other immunoglobulin in the mucosal surface, and also what are the clinical relevance of these molecules in terms of treating some of the autoimmune disorder. So we'll try to move on and see something else in the next class. Thank you.